Hello, I'm really excited to be able to discuss with Dr. ErinHahn her very recent PLOS ONE Lab Protocol article entitled Hot Alkaline Lysis gDNA Extraction from Formalin-Fixed Archival Tissues and the Associated Protocol in Protocols.io. Dr. Hahn is a research scientist at the CSIRO's Australian National Wildlife Collection. She's currently leading projects aimed at developing reliable platforms for airborne eDNA monitoring. The protocol that we will be discussing stemmed from her postdoc at CSIRO, for which she developed methods of extracting genomic and epigenomic data from century-old formalin-preserved museum specimens. Prior to joining CSIRO, Erin completed her PhD at the University of Arizona, where she studied temporal population genetics and epigenetics of the endangered Sonoran pronghorn. Erin is a passionate science communicator and Science and Technology Australia superstar of a STEM alumnus. Erin, thank you so much for discussing this paper with us.
Thanks so much for having me, Gabriel.
And to kick it off, can you briefly explain what this protocol is about, how to use it, and why it is relevant?
So I work in a museum and we have specimens of all sorts of different types and they're preserved in different ways. For a long time, we've been able to get genetic information out of dried specimens, but a whole wing of our collection, those preserved in formalin, have been completely inaccessible because it's been the idea of the field that if something's formalin fixed, you might as well not even try to get DNA out of it. And we had an idea that if we just throw a bunch of stuff at it, try a bunch of different methods and sort out what works, then we can open up those vaults and make them accessible to not just our collection, but collections all around the world. And so this protocol is the result of about 6 or so years of work where we tested a bunch of different methods and found one that works pretty reliably and also gives you a way of vetting specimens that are appropriate for genetic sequencing.
And I hope not to be opening Pandora's box with this question, but for the users who might be willing to optimize it further, you mentioned that you work on this for 6 years, trying many different things, some of which might not have worked. Can you briefly describe what you tried that you thought, oh, this should work, but at the end didn't, with the idea to potentially save some time for future users?
Absolutely, because that's exactly what we wanted, is to save time, researchers' time, as well as saving those precious specimens, because every time you destructively sample, you can't be kind of going into it not knowing if you're going to get DNA out of it. And a few things that we've tried, I mean, we've tried kits, everybody's tried kits. There's FFPE kits for tissues that claim to be able to get DNA out of formalin preserved samples. They don't work very well for museum tissues. And it's not because the kits aren't doing what they say they're doing, it's more about the preparation of the tissue prior to going into the kit. And we've also tried standard proteinase K digestions, which a lot of the kits use and work really, really well on other tissues. But we found that the hot alkaline lysis method that this protocol is presenting works generally better than proteinase K and works more consistently. And again, it's also about that preparation of getting the tissue ready for the extraction.
And I want us to delve a little bit more on what the critical steps of your protocol are. But before to do that, I mean, you mentioned that some kids advertise themselves as working on formalin fixed tissues, and that proteinase K, could potentially work or work in certain specimens. So to better understand the significance of your protocol, can you briefly elaborate on what was the state of the art when you decided to engage in this project? Was there anything reported that other groups have used to extract DNA from formalin-fixed tissues?
So there's lots of work on formalin preserved and paraffin embedded tissues. So the medical field has been doing this for quite a long time, and they're advanced to being able to get lots of different information out of those samples, RNA, doing ChIP-seq, lots of things. And those samples are inherently different from museum tissues because they're very, very thinly sliced, generally fixed with a lower percentage of formaldehyde for a shorter period of time. And our tissues, they're generally full specimens that have been fixed in sometimes 10 times higher formalin, sometimes for up to a century. And so the kits use essentially just heat in an intelligent way in order to decross-link the DNA, extract it, and they don't have to contend with the mass of tissue that's so heavily fixed in museum samples. So they work, and you might get enough DNA out of them, and groups have tried this. There's been previous papers that tried kits. They got enough maybe for some barcoding, doing like very short fragments of mitochondrial DNA. There's been groups that have tried successfully this alkaline lysis method or a pertinase K approach, and sometimes it works, sometimes it doesn't, and it wasn't known yet why it didn't work sometimes, and that's a very critical component of planning your research, is knowing how many specimens am I going to be able to sequence, and communicating that to the museum is also critical. So if you ring up a museum and you say, I want 50 samples, I don't know if any of them are going to work, that's a really quick way to get the museum to deny your sampling request. And so we approached it in a systematic way, comparing methods that had already shown some evidence of working, and we took it further. These previous papers, they'd tried shallow sequencing. They tried doing some form of enrichment. They do capture-based approaches. They'll just do like barcoding. We said, what if you wanted to sequence the entire genome? How hard would you have to sequence in order to get that information? And that gives us a really good idea of the boundaries of what's possible with museum specimens, gives you a sense of how to manage your expectations, and then doing a comparison of different extraction methods, saying which one works in most cases, because museum specimens are highly variable in not just taxa that you're dealing with, but also the preservation quality. And if you're going to have a standard protocol in your lab, you want something that's going to work on most things. And that's why we found that the hot alkaline lysis method, with a couple tweaks that we made on other previous existing papers, tends to work in most cases.
I see. And thank you so much. And now I would like us to discuss what are the most critical steps. Where's those that the user really have to be careful?
Yeah. Step number one is picking the right specimens. There's a lot of specimens in the vaults that are never going to give you DNA. The DNA has likely degraded prior to ever having gone into formalin. You're never going to recover anything from that. And those specimens are better suited for other types of research that aren't destructive and can still give information about that species. So we also tested a number of different parameters of the specimen. So what is the residual formaldehyde concentration in the jar that the specimen's in? What's the pH of the media? And also just what does it look like? And what we found is that formaldehyde level gives you information on whether or not the specimen is formaldehyde exposed. And that's critical because if you have an ethanol preserved specimen, you could use a different approach. And if the formaldehyde concentration is way off the charts, it also tells you that maybe something was done differently with this specimen. It may not have been preserved in the standard approach. And we found a couple of those in the vaults, and they don't generally work. And the other thing was pH is really critical because preservation practices vary wildly, and as over time, and depending on who preserved it, they may have used buffered formalin, they may have used formalin acetic acid, which will destroy DNA. And sometimes we find things in old whiskey jars and we have no idea how this thing was preserved, and the preservation was not recorded. at all. But getting idea of the pH tells you, is it likely that the DNA has been stable since this specimen was preserved? So finding something that's neutral-ish gives you a best idea that that specimen's going to work. But the most critical thing is just having a look at the specimen itself. And if it appears somewhat degraded and the internal organs are not discernible, chances are it underwent post-mortem degradation prior to going into formalin. You're never going to get anything out of that. Just focus on something else. And so we now routinely use this vetting system in the vaults, and it allows us to eliminate those specimens that are just never going to work. And that gets us up to where we were maybe having 10% success of extracting enough DNA for sequencing to our specimens to getting it much closer to 50 to 60, which makes a huge, huge difference, both in your time and the effect of destructive sampling. So absolutely, absolutely check the quality of the specimens, and that requires communicating with the curatorial team. If they can't do it, going into the vaults and having a look for yourself before you send a big long list of specimen requests to the museum. And we're now going through our collection and taking those data so that we can prepare ourselves for when we get requests, we can say, oh, that one's not going to give you what you want. And so let's consider some alternatives. That's honestly, before you even get to the DNA extraction, the absolutely most critical part.
And now that you talk about, generating a database, most of the data that you report, if not all of the data, in your paper comes from tuna larvae. Have you tried, and you've been working on this for years now, have you tried to generalize this to other specimens, to other taxa? And how well does it work? And have you had to troubleshoot or made minor modifications depending on the taxa, more than the quality of the samples, which I understand is very important.
Yep.
But are there difference in taxa?
Yeah, there absolutely is. And we've tried a whole bunch of things. The tuna that ended up in this particular paper, we liked them a lot because they were the smallest specimens that we'd ever successfully extracted. And surprisingly, they yield the highest genomic cover of any specimen that we've sequenced. And we did, for these ones, have to make a slight modification because the protocol itself describes crushing the material using a cryoprep from Kavaris. And for these tuna, they were so small that we didn't need to do that. For some of the larger ones, I went in with a little mortar and pestle and kind of just ground them up a little bit. But putting them into the cryoprep was kind of pointless. They were so small. So for those ones, we used the lysis buffer, just popped the whole specimen in, which was really handy because it removed a step, but that's something that you could only do with a very, very small specimen. We've also successfully extracted DNA from a whole range of eastern water dragons, which are a native species here in Australia. We've, from that species, extracted DNA and resequenced the genome of our oldest specimen from 1905, and that one gave us up to about 8x cover. Which will be in a subsequent paper that we're putting together right now. We also have recovered up to about 4x cover for a cane toad, which some have quite large genomes, which is quite a feat. We've done amphibians, we've done reptiles, we've done fish, we've done birds, we've done mammals, and in terms of vertebrates, there isn't much variation in what you have to do with the protocol. Tissue's kind of tissue. Some tissues are a bit tougher than others. We've primarily worked with liver for all of our specimens, aside from this teeny tiny tuna, which were done whole. We generally go for liver because the specimens that we're working with are larger and they generally have an incision down the median of their body. The liver's sitting there handy. You don't have to dig around into it and get it. Generally works better than skin because there's just more connective tissue and fat to deal with and it doesn't extract as well. We've quite successfully sequenced ovaries and testis from other tuna as well. And We've done skin on a few, and we had a paper that's currently in Molecular Ecology Resources, published in 2022, comparing a whole bunch of different taxa as well as different tissues. And in that particular experiment, for those that had liver, we extracted liver. For those that didn't have liver, we generally went for skin or some muscle tissue. And the muscle and skin didn't work very well. However, we think it's because for those ones that didn't have liver, they didn't have liver because their entire gut cavity had been removed, which indicates to us that there had been extensive postmortem degradation. They removed all of the stuff that had started to turn prior to putting it into formalin. So for those ones, it's more of a reflection of quality as opposed to tissue. And so now we're looking at it as go for liver if you can, because it's easy, and otherwise something that has a little bit less connective tissue if you're going for genetic sequencing.
Thank you so much. I think this information is going to be very useful for the future users. And I would like us to change gears a little bit and talk about the peer review and to begin with your motivations. I mean, you work on this for 6 years. You put it on the open access platform about 6 months ago, but then you went a step further and decided to get this peer review. What was the motivation to send the protocol to a journal?
So our paper where we did the comparison of this method to other ones was really well received, is getting lots of citations. We get questions all the time for clarification on the protocol. And as much as I really want to be answering those emails, it would be most effective for me to be able to point people to a protocol, as well as include updates. So in the original paper, we didn't use a bead-based purification of the DNA. We now do that. So I included that in the paper, as well as an optimized solution of our SPRI beads, which really focuses on getting those really small fragments. And we also included information about limitations, how to manage your expectations of what you're going to get out of these specimens. So before anyone touches a specimen, they could read this protocol and know whether or not they're going to be suitable. So that was really our driving motivation, to put something out there that's a step-by-step bench protocol. And we also then wanted to make sure that we were pointing people to it in a way that they could cite it, that they could engage with the literature. And that's where I came across the option to couple that that protocol with a PLOS ONE Lab Protocol, and it seemed absolutely ideal to get a little bit more attention drawn to that protocol so that people could find it and engage with it.
And how did the feedback that you received from the peer reviewers shape the final version of the protocol?
The peer review process was really great. It's fast, and they were also very helpful in making sure that it clarified all of the necessary points. So in the protocols IO version, I had a whole bunch of notes in the top, which were those managing your expectations. Don't expect to be able to do de novo sequencing on these. You're going to get 65 base pair fragments. You're never going to recover the whole genome. You have to have a reference. If your sample is poor quality, at most you can expect to be able to get a mitochondrial genome. Don't put all of your eggs in that basket for like a PhD project. So all of that stuff was in the notes for the protocols IO. And when I was writing up the paper for PLOS ONE, I had neglected to infuse that into the paper itself, thinking that people would look at the protocol. And the reviewer very helpfully pointed out a few areas where I could help drive home those managing of expectations, which then prompted me to put an additional paragraph and a half or so, really driving it home so that people, again, don't waste time and resources on specimens that are never going to work. So I found that really helpful.
And what was your motivation to share publicly the peer review history together with the paper?
A couple things. I believe strongly that people should be acknowledged for their contribution to the peer review process. It's time consuming, and people put a lot of heart and soul into it, and we should be acknowledged for that work. And then also, I think having it being open makes people a bit more collegial. If you know that your name's going to go on a review, you're going to make sure that that review is as helpful as possible and friendly and collegial, because that's really what the process should be. So that's why I opted to make that process entirely transparent.
And from what you have described already, it seems that the development of this protocol has had an impact on the field, and people are engaging with them. They are contacting you. So I hope that this the protocol and the relatively recent paper, continues that trend of engaging people with your methodological developments. And I'm really happy for that. Before we finalize, is there anything that you would like to add to mention that we haven't touched so far?
I think some further optimizations of the protocol could be done, especially if you want to start using this for plants. There are some formalin preserved plants. I would not jump in and do this protocol for plants because I'm a vertebrate person. I don't know the first thing about plants and cell walls. The key components of it, heat, and really disrupting the tissue, all of those could stay the same, but with some modifications for all those things that plant people do. So that's one thing. Another thing is some of the parts of the method, if you don't have the specific equipment for it, so if you don't have a cryoprep, just crushing the tissue in some way is absolutely better than nothing. So I wouldn't use a tissue lyser. We did try that. It seems to be a bit overkill to get it so fine. It does tend to destroy the DNA a little bit, but just something to break up that tissue would really be very much better. And there's also the use of phenylchloroform. We use it. It works beautifully. It's something I'm very much accustomed to using. It's a skill in and of itself to properly do phenylchloroform transfers. And if you haven't done that before, absolutely have someone who has experience with these to show you how to do it. There's also the option for trialing methods of purifying the DNA that aren't reliant on phenylchloroform, which is something that we're currently exploring. But I think that that's another area where your lab could try different things, especially if you know how to use alternative methods.
Erin, thank you so much. It's been a fun conversation. I think it's going to be very useful for the users of the protocol. And with that, just to thank you again.
Yeah, thanks so much. I hope it's been helpful.